Thanks for coming. Basically, we use all sorts of genomic information to construct a 3D human genome model with all 46 chromosomes in a 3D space. And the model contains all the useful information about nucleosome position, the sequence, and atomic structure, and things like that. So this is my topic today. So this work is based on this paper we published last year in the BMC Bioinformatics, a Genome3D, a viewer model framework for integrating and visualizing multi-scale epigenomic information within a three-dimensional genome. And this is a work between my group and Dr. Jijin Tang's group at the University of South Carolina. So first I will talk a little bit about the introduction and background, and then I'll talk about the 3D genome model we constructed, and then I'll talk a little bit about the viewer software for the 3D genome model, and then I'll talk a little bit about the applications. As we all know, we got a human genome sequence, 3 billion base pairs, but getting sequence is only the very small step toward understanding the genome. We know that besides the genome sequence, we also have lots of other informations. For example, the nucleosome position, where each nucleosome located on the genomic DNA. And there's also the modification, DNA methylation, and histone methylation, and how those play biological role is still under study. And then there are information about how the genome function. For example, the publication shows that the actively transcribed genes form sort of a transcription factory, where all these genes gather together when they need to be transcribed. So this sort of structure view is very difficult to understand just by looking at the sequence itself. And in addition, there are new technologies that start to look at the structure aspect of the genome. For example, this work is by Genome Institute of Singapore. They use what's called chromatin immunoprecipitation combined with paired and dye-tag sequencing to study the genomic elements that are in close proximity to each other. And then there's this high-C method, which taking a similar approach using the cross-linking combined with the high-throughput sequencing to study, again, the proximity of genomic elements in the three-dimensional space. And people also apply the similar technology to study the three-dimensional model of the yeast genome. So with all this information, it is sort of make people think, or at least make me think, that whether it is possible to build a 3D physical model of human genome. And certainly there are more and more data become available. For example, a work from Dr. Keji Zhao's lab from NIH, they use chip sequencing method to determine the precise location of each nucleosome in certain types of cells. And also, there are a lot of work from the ENCODE project to study the DNA methylation, histone methylation, and things like that. And certainly, as I mentioned, there's this high-C and chair-packed method to determine the contact points of genomic DNA in the nucleus. So, with all this information become available, and also, as we all know, we are getting faster CPU, faster GPU, faster memory, faster chip storage, such as solid-state hard drive. And, with all this hardware advantage, we might also be able to visually display and process a 3D physical model of a human genome. But there are still a lot of challenges. For example, if we want to build a human genome down to the atomic level detail, if you think about it, we have 3 billion base pairs. Each base will have 20 atoms. And then for each atom, you need to give 3-dimensional coordinates. So that's 3 positions. And then for each coordinates, you need 4 bytes to store the data. So if you calculate all of those, that's 600 gigabytes of data. Okay. And so the challenge then is how to integrate and visualize this complex, vast amount of data on your desktop. So the approach we've taken is we use different measures to build a 3D model of human genome. So here are the data sets we used. There are, genome sequence, that's at the atomic level. And then there are chromosome positions, 30 nanometer fiber pad flexibility, and the nucleosome base pair positions, and nucleosome core particles. These are the protein structure information from PDB. And also tetranucleosome array is, again, also structure level data. And then we also take some information from genetic studies for the single nucleotide polymorphism, and also the histone methylation information from ENCODE project. And then we also use the base pair atom positions. So we're taking all these data, and then we take we take all this data to model the interface chromatin in a 3D space that is comparable to the size of the nucleus. And we build this model at 4 different scales: the nucleus scale, the 30 nanometer fiber scale, the nucleosome scale, and the atomic scale. And the model integrates multi-scale data to realize a full atomic level details. For the model construction, we take a top-down model construction approach. Each level uses the data from a higher level or starting point. So, for example, we start with the nuclear level, and we construct the model, and then we use that model to constrain the next level, which is 30 nanometer fiber. And by doing that, we basically ensure a consistent model across all levels. And also, the anchor points from higher level constraints the lower level scale structure, so that is consistent again. And then we use a 3-dimensional curve, basically the catenary-Rom spline, and constrain the random walk for the model construction. So this is just a simple, I'll give you a simple idea about the random walk. Basically, you have a cubic, lattice cubic in a 3-dimensional space, and then you can take n steps to walk through this cubic space toward the next goal. And then we use a function to constrain the random walk. The details can be seen in the paper we published. And then we also try to manage the data size. As you can see, I said that, combine all the data, it's 600 gigabytes, so that's humongous. So we use different approaches. Here's one example of that. So we know that, the nucleosome structure is pretty consistent. You have about 147 base pairs wrapped around the nucleosome structure. And so if you calculate the size to contain all the data, it's about 32.58 kilobytes. However, because nucleosome has a consistent structure, instead of we keep track of all the atoms in the nucleosome, we can simply take the center mass of this nucleosome, and then we calculate the whole nucleosome structure on the fly. So that reduces the data size to the 12 bytes. So here is basically we use a divide and conquer approach to model all these different levels. The top is the whole 46 chromosomes, the human chromosomes in the 3D space. And then the bottom left panel is a 30 nanometer fiber And then in the 30 nanometer fiber, you might be able to see those loops, which are the actually nucleosomes. And then you can get down to the atomic level to see the individual atoms in each base from the sequence. So we built a model. Certainly you need software to visualize that model. And so we also built a view software for 3D genome model. And here is a 4 different level view from the software viewer. So in the panel A, that's at the nuclear level, with all these chromosomes are roughly displayed in their position. And then in panel B is a 30 nanometer fiber level, where you can see that all these chromosomes in the 3D space with individual chromosome occupy its own space. This is using a known knowledge about the distribution of the chromosomes called the chromosome territory. And then in panel C, you see the at the nucleosome level, you can see DNA wrapped around the individual nucleosome and in the 3D space. And in the panel D, you see the atomic structure of an individual nucleosome with all the atoms of the DNA and the protein can be visualized. And in the software viewer, we also implemented something called a gizmo. And what it is, it's a sphere structure that you can control its size, and then you can move this around. Whatever that's included in this sphere structure then can be visualized at a greater detail in a different panel. So this is very convenient. If you find some genomic feature that's very interesting, you can simply move this gizmo into that area, magnify it, and visualize the details in another panel. So in summary, the Genome3D viewer is very flexible. It can visualize other models. For example, there are other groups that are developing, also Genome3D model, as you see from the second talk. And so our software viewer actually visualize other people's models as well, as long as the model is specified in format recognized by the software. And also annotation capable. So we can annotate all these elements in the genomic model, especially structure-related features. And it also works with the UCSC Genome Browser. So you can use the UCSC Genome Browser, extract genomic elements with the features, and then you can import those data and annotate the model using those data. And so you probably ask, what's the use of these? So I'm talking a little bit about application and analysis. The first one is about the location, the structure-related location in the context of nucleosome. As you know, the DNA wrapped around the nucleosome, and also the DNA is a double helix. So from this picture, you will see that when DNA wrapped around the nucleosome, the phosphate in this major and minor groove, which is important for the protein-DNA interaction, they have different phases. Some exposed to the outside, and some is buried between the histone and the DNA strand. So this different phase is very important for the protein-DNA interaction and for the function of some DNA elements. That has been demonstrated by previous work. So obviously, if a base with its phosphate exposed to the outside, that can be easily accessed by the protein that binds to the sequence containing that base. And if it's buried between the DNA strand and the histone protein, then it's not easily accessed to outside protein. And then this phase information might, influence the function of that particular base or the sequence associated with it. And so using our viewer and our structure, we can sort of look into that information. So here are the 4 SNPs that's identified that may associate with the risk of cholesterol level from a genetic study. So these all 4 SNPs are all within a particular nucleosome position using the data from Dr. Keji Zhao's lab. And you can see that these SNPs 1 and 2, they are pretty much buried between the DNA strand and the nucleosome, where the SNPs 3 and 4, they are exposed to the outside. So this kind of information can give you some indication about which SNP might be functionally relevant to the phenotype. This might be particularly useful for SNPs, for example, in the non-coding region, where it's very difficult to figure out their function. Where using this sort of structure feature, you might be able to prioritize these SNPs for the further investigation. So another example is because our nucleosome is based on the protein structure of the nucleosome complex, we can actually highlight, for example, the methylation of histone with these yellow dots. So with these kind of information, we know that histone methylation is important for the gene regulation. And with this kind of structure view, we hope down the road there will be sufficient data sort of allow people to figure out how this histone methylation might interact with other machinery in the genome to influence the genome function. And we can also use this as a platform to visualize large data sets. For example, here is a visualization of a genome-wide gene expression in response to p53 together with the p53 binding site information. So basically, one data set is from experiment. People knock out p53 and study which gene up and which gene down. And the other data set is study where p53 binds in the genome. And from this figure, you can see that the orange cubic, for example, like here, is a p53 binding sites that has a gene up or down regulated within 10 kb of that particular gene. So, which means this binding site probably is a true p53 binding site with a biological consequence if you delete the p53. Where the yellow cube are the p53 binding sites that, it does not influence any gene expression. And you can visualize the whole chromosome in one picture. You can rotate, you can zoom in, zoom out in our software viewer. So it's an intuitive interface to visualize large data sets. There are also other related works. For example, this group, I guess the second speaker, they use the high-C data, try to construct the 3D dimensional folding of the alpha globin gene domain. This is another group, use this 3D proximity of evolutionary breaking points, argue for the notion of spatial synteny. So basically, the genomic region with close contact to each other, they are easier to break and the translocation is easier to occur. So there are certain caveats about our model. First, we use approximations because they're not, sufficient data to precisely define every aspect of the model. And the spatial distribution of chromatin is statistically derived, constrained on known biological knowledge. And we are in the process of model refinement using recent 3D structure data from chip DITAC, pair-end DITAC sequencing and high-C. And certainly, what we have shown here is 3D genomic model is doable, but biologists, please provide me with data to make it accurate. There are also caveats about Genomic 3D viewer. This is a standalone Windows application. It's difficult for data sharing and collaborative work. And we are planning to develop a web version so that, it can facilitate data sharing and collaboration. And more additional detail will be presented at post J50 on Tuesday, tomorrow, from 12:40 to 2:30 in the afternoon. And here's the paper, software, or you can just simply search Genome3D. It'll get you to the download page with links to papers and software and model. And so I think one thing I really want to point out is that genome structure in 3D is a new area of research with a lot of opportunities. It's a new angle to look at the genome, and there are a lot of new data that can use for this. And also then it's a new research front, data integration and visualization and data analysis with genome structure feature and also algorithm development. Certainly there are new applications. One thing I want to mention is, for example, there are a lot of talk about the personal genome information. So how do you expect your grandparents to go to UCSC genome browser, find out their SNPs? It's difficult, right? But with a graphic view that, you can rotate, zoom in, zoom out, maybe potentially on your cell phone, it might be a useful interface to deliver personal genome information. So I just want to acknowledge Tom Asbury in my group and Matt Midman in Dr. Junjin Tang's group are the major contributor to the model and the viewer. And Tintin and Alex in my group did a lot of testing. And we appreciate that Dr. Keji Zhao's group provided the nucleosome position information. And Yijing Ran is a former co-worker at the Genome Institute of Singapore. We had discussion about this project at the early phase. And Dr. Mike Benke also read the manuscript, and Kelly did a lot of testing as well. And the funding agencies provided support.
Thanks a lot.